I'm your host, Dr. Catherine Clinton, and today I am honored to be here with one of my idols, actually, in science and medicine. Dr. Jerry Pollack is here with us, and he is a professor at the University of Washington. He is a prolific author with research articles. He wrote Cells, Gels, and the Engines of Life. He also wrote The Fourth Phase of Water, some very popular books out there about the new science behind our biology. He is the founding editor-in-chief of the Water Journal, which is an incredible resource for those of us wanting to learn more about water and its properties, both in physics and biology and engineering. And he also hosts the Water Conference annually, which is another wonderful resource. He puts up talks on YouTube. He has so many articles out there, and I am just absolutely honored to have you with us today. Thank you so much for taking time out of your busy schedule to talk with us.
Oh, thank you, Catherine. It's my great pleasure to be there with you. Um,
Wonderful. Well, before we get into some of the nitty-gritty around the science that you're doing, tell us a little bit more about this fourth phase of water. So this is something that um, I was introduced to maybe 4 or 5 years ago in the research, and it really, blew my mind. It's sort of that education we didn't learn in school, right? And many of us think of H2O or water as this sort of static thing that is just uniform, and it's that oxygen with the little hydrogen Mickey Mouse ears in liquid form. and and really we now know that it's much more dynamic, right? Those hydrogens are moving and dancing in such a fluid way. And you and your research lab have really teased out some really important properties of this fourth phase of water, this exclusion zone water, structured water, ordered water. So before we dive into some of the nitty-gritty pieces around this, can you explain to us what is the difference between that H2O water that we're drinking in our glasses and that water within our cells, that structured water?
sure. Where where to start? Um, well, we all learn as students in middle school that water has three phases: solid, liquid, and vapor. Everybody knows that. And what we discovered really does, or what I should say, identified, is a phase beyond that. And it's not just a trivial laboratory phenomenon that you could see under certain odd circumstances. This water is basically all over the place, and it fills our bodies. And the way you can quickly demonstrate that it feels that our bodies are not filled, it's not liquid water, just cut yourself. So if it were liquid water and you cut yourself, it would come pouring out like a breached pipe, water-filled pipe. That doesn't happen, you see. So it's got to be different. And long ago, this was 60, 70 years ago, there were studies by a German, or maybe he was Austrian, Frei Wüstling, who wrote a whole book providing evidence that the water inside our cells, pretty much inside our bodies, is gel-like. And so we don't the three phases that we talk about don't discuss any gel-like water, but gel-like water, actually, the gel is the actual consistency of this fourth phase that I'm talking about. Now, this wasn't just pulled from thin air. There had been actually a bunch of studies early on, or I should say a bunch of people, some of whom rather distinguished people, who knew that the water inside the biological water, shall we say, differed from ordinary liquid water. there were two in particular. One of them is Albert St. Georgi, who you know, but I'm not sure. You know, it's been a while. So he's a Nobel laureate. Was a Nobel laureate. He discovered vitamin C, and his mind was so fertile. He was involved in many different fields, but he also was involved in water, and he knew that the idea that the body was filled with liquid water is not correct, that it's a different kind of water, although he didn't identify it, but he knew about the centrality of water, that the water is not just a solvent that in which the most important molecules of life were sitting, and didn't really do a whole lot. He said, no, that's not the case at all. And one of his famous aphorisms is, life is water dancing to the tune. Oh, you know this one, dancing to the tune of solids. So water, he said, and he had evidence that the water played an integral role in everything that the body does. And then came, there was some overlap between the two. A guy named Gilbert Ling, whose name you probably know, but I'm not sure. He's not as, you might say, as well recognized or as famous, but Gilbert Ling studied water and biological water. And I should say, in terms of background, that he was almost, you might say, as distinguished as St. Georgi, except he never won a Nobel Prize. He came from China. He was in the first cohort of scientists chosen from throughout China to come to the US to study, young, intelligent, brash, promising young scientists. And of the three, one was a biologist, that was Ling, then there's a chemist and the physicist. And the two latter ones, in their respective fields, went on to win Nobel Prizes. And Gilbert Ling did not win a Nobel Prize because he was too controversial, but he certainly deserved at least one and maybe more. And his main theme was that the water molecules inside of the body were organized. He called it structured water. And I mentioned these two because it's on the shoulders of these giants that we began to build. The idea of a different kind of water was not pulled from thin air. So I met Gilbert Ling at a conference. I was charged with presenting material at this conference in the field of the molecular mechanism of muscle contraction, in which I was involved for a couple of decades. And Gilbert Ling and others were invited to present their work on water. These two topics were of interest to a biophysicist who had died, and the conference was in his honor, a memorial kind of conference. So I presented my stuff, and I don't know if it was well received or not well received, but Gilbert Ling presented, and I was astonished by what he was presenting. And I was astonished even more by the dozen or so people who had evidence to support his point of view. I was hooked. And so I went back home, and I was just so excited about it, almost ready to change fields, which I did. And I gave one of his books. By that time, he'd written like 5 books. And I think before he died a couple of years ago, I think there were 7. And in most of them, he was talking about and presenting evidence for the existence of this so-called structured water. So I gave a book to some of my favorite students, and they got back to me, and their comment was, boy, this guy is on to something critically important. And if they're right, it changes everything. And so I was buoyed by the positive responses, and I thought, okay, what are we going to do? I'd been in the field of muscle contraction for a while. There was more to contribute. I made my contribution. Almost nobody paid attention to it. Although I think the arguments still hold that the prevailing point of view is not correct, and there are other ideas involving water. Because water, 2/3 of our volume by water, by volume, 2/3 of our volume is water, right? 2/3. And if you translate that into a molecular count, you line up all the molecules. So more than 99 out of 100 molecules are water molecules. And the idea, the concept, which is prevailing in all of biology and medicine, is, yeah, big deal. Those 99 out of 100 molecules don't do a whole lot. They just kind of sit there to bathe the more important molecules of life. This didn't seem rational to me. So anyway, I abruptly changed course, and I hope the NIH administrators are not listening, because I had some money to study muscle contraction. I took that money and shifted it to begin studying water. And within year one, we already had evidence. And so it turned out, without going into great detail, it turned out that our evidence showed that Gilbert Ling was on track, but not exactly right. His idea was that the water molecules, being dipoles, as you mentioned, the Mickey Mouse ears, the positive and the other end negative, the dipoles plus minus could line up, as you could imagine, minus plus, then next one minus plus, and so on. That was his idea. We found that the idea, although the general idea of order was absolutely correct, but his idea was not tenable, because we found that this special kind of water typically bore negative charge. Dipoles don't do that. They're neutral. They've got minus at one end and plus at the other end. So you can pile up water molecules from here to the moon, and you'll never get negative charge. But we measured directly using electrodes, parenthetically invented by the same Gilbert Ling, that showed negative charge. So it had to be wrong. And, I was not happy that Gilbert was wrong. And the first book that I wrote, you mentioned the name Cells, Gels, and the Engines of Life. At that time, we didn't know. That was my first effort. And the effort was directed toward making Gilbert Ling's ideas, shall we say, understandable to the average non-genius, because Gilbert Ling would sit down at the typewriter or the word processor later and bat out something, send it to the publisher, who would print it, put some covers on it, and that was it. You can't write a book that way. Maybe you can, because you're so articulate. You've got to go through and edit and get criticism or whatever. So anyway, it was my attempt to make his ideas understandable. He was not happy with my book. He was not happy with it because it didn't mention his name enough times. And when you write a book like that, the first half of the book was about Gilbert Ling and structured water and his ideas. And you can't mention the name Gilbert Ling every paragraph. But I thought, it was dedicated to him and such. Anyway, and I went on to show that an old idea probably had some merit to it, some truth. The idea that when the cell is full of this stuff, when the cell goes into action, for example, when a muscle cell starts contracting or a secretory cell starts secreting or whatever, part of the process is a transition of the water from the structured water that Ling was talking about to ordinary liquid water, and then back again when the event was over. And whatever, this was the beginning. And then after that contribution, I thought we really need to study the water experimentally. And that's where we began the experiments that I talked about, including sticking the electrodes in to find that the water was negatively charged. So that's how it all began. And I just want to emphasize that the concepts, ideas, hypotheses were not pulled out of thin air. They started with people like Ling and St. Georgie and others who I don't have time to include. There were many people who really understood that it was not simply the H2O molecule, but a lot more than that. And there was something that looked, in biology especially, and we actually found it goes beyond biology. It's everywhere. This kind of water that we called fourth phase water, or EZ water, EZ standing for exclusion zone, because this water formed a zone, and we found that it excluded just about everything. As crystals, this is a kind of crystalline water, as crystals do. They just like ice is a crystal, and to get pure ice, the water that formed the ice may have had impurities, but they get pushed out in order to form a pure crystal. And so it's the same with this easy water. And I think I better stop and let you ask questions because I'm rambling on and on.
Well, I really appreciate your acknowledgement of the people that came before and the scientists that were doing this research before, because, you know, it's something with when we're talking about quantum biology, you can come to a point where you've come full circle and you're talking about these ancient medical practices that might be utilizing that structured water, right? So some of this stuff might even go back even further than Ling and some of these researchers that you're talking about. But I do appreciate that acknowledgement, because sometimes, oftentimes in science, we don't get that acknowledgement. You know, people like to to have the spotlight. But Jerry, can you tell us a little bit more about those first experiments, those first few years where you are finding that structured water against hydrophilic surfaces, where you're finding that it creates a battery, where you're finding that it acts different in these hydrophilic tubes? Can you explain to some of our listeners today who might be tuning in and hearing about structured water for the first time?
Absolutely. Oh, okay. Yeah. And remind me if I get off track about the tubes, because the tubes are really important. Well, we started the experiments building directly from Gilbert Ling. So Gilbert Ling's idea was that the molecules were organized, and it forms a crystal. And as I mentioned, crystals tend to exclude what particles, molecules, and whatever. And I had the good fortune of running into a couple of people who provided an impetus to go experimentally in a certain direction. So here's what we did, and from this, the properties will come out naturally. So we took some water and we put some particles in the water, little micron-sized spheres called microspheres. They're commonly used experimentally. And into that suspension of spheres, aqueous suspension, we put a gel. The first one was it doesn't really matter, it was a polyvinyl alcohol gel. We put it in the water and we looked in the microscope to see what happens. And we found that right adjacent to the gel, which is a hydrophilic surface, that is water-loving surface, we look in the microscope and we could see progressively those microspheres began to be excluded from the region next to the gel. And so we called it exclusion zone because it seemed appropriate. And it was, in some ways, it was astonishing because it would grow and grow and grow, and we in fact didn't even need a microscope to see it. It would grow typically a few tenths of a millimeter, sometimes even up to half a millimeter. Later we saw even up to a millimeter, and actually in certain circumstances even beyond that. So we thought, my goodness, this could be what Gilbert Ling was talking about, the somehow. And then we began looking at the properties of this, and we found a whole lot of interesting features. First of all, we and also another group, a Russian group, found that the water in this region, this easy water. Later we called it fourth phase water because it was so different from it, from the liquid phase, that there was order to it, a lot of order. So whatever was making up this this region showed a lot of ordering. And we also, as I mentioned, found or did I mention, a negative charge in this region. And in fact, we found beyond that region was an equal amount of positive charge. Why that? Um, well, it all starts from the water molecule, and so the water molecule is neutral. And if you have a region now um, that's negatively charged, you need to find a region that's positively charged, because the positive charges, they they're there. So, and we found that, and what this constituted effectively was a battery, because you got a negatively charged region sitting next beyond to a positively charged region. And we thought, this is pretty interesting. So if we were to stick one electrode in the negative and one electrode in the positive, we ought to get electrical energy out of it, because just like a battery. And we did. So it was enough. We have some videos that we show, enough to light a light bulb, or to an LED, I should say, at least. So it wasn't a phenomenal amount of energy, but the principle was there. So next point, you can't get something for nothing, right? And so if you have electrical energy potentially that's coming out of it, you need something to charge up that battery, and also something to create the order. If you go from chaos to order, you need to put in energy. It's like cleaning your office. It takes some. It's easy for it to become chaotic. You're busy doing something, you plunk down something, but putting it back in order requires some energy on your part. I don't know how often you do that, but maybe you do. So where's the energy? And I got to tell you, we spent a couple of years in head scratching. Where on earth does this energy come from? It was not obvious, because you can't take the chamber and plug it into the receptacle for 110 volts. That that doesn't work. Okay, and so where else? Well, so we wound up. Well, I should say, it was a student doing an experiment that he was not supposed to do, who taught us where it's coming from. So he's doing the experiment that I was talking about, putting a gel in the water with the microspheres, and I guess he got bored or something. So there was a lamp sitting right nearby, and he took the lamp and shined it on the chamber, and where the region that was illuminated, the exclusion zone, that several hundred micrometer region, It grew enormously. And he called me in and he said, "Hey, take a look at this." And immediately it was obvious the energy was coming from light. So I asked him, I said, "Why don't you turn off the light?" And sure enough, this region that had grown enormously went back. It took tens of minutes to go back to the original. So then we did experiments, and we wanted to know which wavelengths of light, and we found it was not the visible ones. It was not the ultraviolet. It was infrared. Infrared energy is, and we with very weak infrared energy from an LED, we could obtain a growth on the order of 10 times the exclusion zone. Really powerful. Very weak light was sufficient to grow this. So so I guess so I don't ramble on some more. I think those are some of the fundamental properties of this zone. Now, you did ask me, and I did remember about the tubes. You want me to talk about the tubes? Because
I do. I want you to talk about the tubes, but I want to just take a step back for
I thought that might be the case. Go ahead.
for everybody listening. So when we're talking about ordered, aligned, we're talking about the molecules in the water, right?
Yep.
Okay, and that structure creates this hexagonal lattice formation of the hydrogen atoms within that structured water.
Hydrogen and oxygen, right. Yeah, I neglected to mention it. Thank you for the reminder. Yeah, this region, this several hundred micrometer wide region, we found, as you mentioned, it's actually a hexagonal sheet structure. So next to the hydrophilic surface, the gel, but it doesn't need to be a gel. It could be polymer or many biological surfaces, many surfaces, as long as they're hydrophilic. So what happens is that the water molecules undergo a transition and they morph into a sheet-like structure, which if you look at it, it looks like a honeycomb, a bunch of hexagons. And once you form the first layer, the first layer forms a template for the growth of the second layer and the third layer, and they keep growing layer by layer. And there are many layers. We're talking about hundreds of thousands of layers easily. And that's the structure that we're talking about. It's an ordered structure consisting of hexagons, and one hexagon builds after the other into a pretty extensive we say easy water region or fourth phase region. Okay, so go ahead. Yeah.
Now, this fourth phase of water region, this easy zone, this exclusion zone, you put an electrode in that and in the accompanying bulk water that
Yep.
forms outside. And that energy created by the relationship between those two is what powered that LED light.
Exactly. Thank you for clarifying.
Just absolutely incredible. Now, I really do want you to talk about what's happening with those hydrophilic tubes because uh, it's absolutely fascinating and really important when we start talking about quantum biology and how things are being moved throughout living systems.
I will, but let me first um, just um, digress just a moment, because maybe it's not obvious, but this battery like feature, it's involved in our cells, and it's a form of energy, and if you make the connection between that and energy supply, um, this fueled by infrared energy, and there are other ways too. It's a transduction of infrared energy into electrical potential energy, which can drive all kinds of processes in biology. So I just wanted to make that. I wanted to make sure that I would mention that, because it could be really important, because obviously in order to do what we do, you need energy, and we think of the energy only as ATP energy, but this energy, we don't know how much of the energy is electrical, but at least some of the energy is electrical. Okay. That
Absolutely. Absolutely. That was you had your Gilbert Ling moment. I had my Jerry Pollack moment where my mind just blew and I was hooked. And that's what happened when I read about your research and what you were doing. It's absolutely phenomenal. And I do think that the evidence points to how important this is in living systems. So thank you for pointing that out. And
okay.
let's take it further and talk about those hydrophilic tubes and what
Yep.
happened in your lab with that.
Okay. Well, this was another experiment um, by an undergraduate student who it was serendipity, but meaningful serendipity. So we found at first we found we used hydrophilic surfaces. It's called Naphion, and it's a how should I say? it's a polymer, and it's easy to work with. It's right there. You cut a little piece, and you do experiments. And then we found that it actually comes also in not only in sheet form, but also in cylindrical form, like a tube, like a straw. And so I thought, well, might as well experiment with it. And I asked a competent student, I said, this thing has it's not linear, it's circumferential, and please, can you determine whether there really is still an EZ? We knew that there's an EZ in the flat surfaces, but we didn't know because of the curvature whether, and he found immediately, no problem, outside and inside, there's an EZ that forms. And I was busy doing something or other. I'm not sure whether it was going to Europe on a seminar tour or whatever, but he didn't have anything to do because he already figured out, and so he kind of fumbled around and one day he came to my office, not knowing exactly what to do, and I was busy talking to somebody in my office, and as I recall, it wasn't the most stimulating of conversations, and so the interruption was okay. So he came, and I remember I left the door ajar, which is what I often do, and he opened it, came by, and kind of brashly said, I think I saw something interesting, and I thought maybe I'd like to tell you about it. It was slightly impolite because I was speaking with some guy who, as I recall, was not necessarily the most stimulating of people, and so but so I was happy to hear about what he had to say, and I thought he said to me, he said, the water keeps running through this straw-like configuration. And I'm thinking, if this is right, it's going to be really interesting. And it turns out it was right. I went into the lab and determined that this is real. And I thought, this is more than just it's astonishing because in order to, if you think about it, in order to let water go through a tube, the water has viscosity to it, and in order to the water to go through the tube, you need energy. So where does this energy come from? And I thought we had just found out that infrared energy would somehow build EZ. And so I was thinking, well, maybe it's infrared energy, and sure enough, that's what happened. So we built many different experiments, and we found out, without going into great detail, that indeed it was the water that was going through, and it was based on infrared energy. And so, just to make the story a little bit more complete, the energy builds EZ water, annular EZ water, just inside the tube. And at the core of the tube is protons, is positive charge. So you have, again, as usual, negative charge around the periphery and positive charge. And those positive charges, what they are essentially protons that are free. And so, since they're free, they start building up, and they want to escape. Since they're free, they'll either go to the left or to the right. And we found that indeed they go. We're still not exactly sure about the directionality, but once they go, so for example, once they go in this direction, once the protons go, and the protons are attached to the water, so you got positively charged water that goes, and new water gets pulled in from outside, and the positively charged water builds again, and it just keeps going on and on. And so we found that in the laboratory, they could keep going for as much as a day and a half, and we now know it could go indefinitely. We understand why. So this is really interesting. And then the second part of this, if I explain so far what's going on, I want to talk about the second part because the second part involves blood, and I think you know what I'm talking about. This is an experiment. I went. It was a trip that I experienced going to Russia, and it was just after we found about these tubes and the protons that move through the tubes. And I was visiting my friend Vladimir Vyakov from Moscow University, and quickly he introduced me to a neighboring laboratory. And the guy was telling me there's a big problem in the cardiovascular system. And I must admit, I was skeptical. It was like, I had my nose up in the air because it's sometimes called cutting the clouds with your nose, a sense of arrogance. Because in my graduate studies, I had studied the cardiovascular system. I'd studied the pressures and flows, and I thought we had it worked out. Well, after 5 minutes, I came to realize that he was right, that there's a big problem in the cardiovascular system. So what's the big problem? The problem is very simple. The problem is that the red blood cells are 6 or 7 micrometers in diameter, but people who are healthy young adults like yourself are maybe 3 micrometers, so half the diameter. So this is a problem. You might say a plumbing problem, right? Because you have these big lumping blobs trying to get through these narrow tubes. Somehow they manage it, but in order to do it, what happens is that they squeeze each of the red blood cells, and in squeezing it, they narrow their diameters, and they somehow manage to get through. And you could see the evidence. I typically show a video. You could see that they get squeezed, but in order to get squeezed, it requires a lot of energy, right, to do it. And this Russian guy, calculated how much energy is required to squeeze every one of those red blood cells. He computed that an enormous amount of energy, because you can imagine, a lot of red blood cells. And his computation was that if the left ventricle was generating enough pressure, the amount of pressure that would need to be donated is something like 1 million times the actual left ventricular pressure. Amazing. So even if he's wrong by 1 or 2 orders of magnitude, which is possible, still it's an enormous amount of energy. So there's got to be something else. There must be something else that is powering the flow of erythrocytes or of blood. And so we decided to look into this in more detail, and we tested to see whether there is another driving mechanism, the signature feature, okay, which is adding infrared energy. So we added infrared energy, and we found that the flow could increase by 3 times or more just by adding infrared energy. So we tested the signature feature of what's going on, and it didn't prove that this is the mechanism, but it's certainly compatible with the idea that the driving force is essentially infrared energy that involves the easy water. And so the bottom line is that it looks as though blood flow is more than just the manifestation of energy from pressure. There's something else that goes on, and this is the flow that gets driven through these tubes, known as the capillaries and red blood cells. So this is exciting for us because it looks as though what drives the blood is not just left ventricular pressure. That's part of it, but it also involves um, an adjunct, and we don't know which one or how much is being driven. It's the vessels themselves that drive through, and we don't know whether it's the minority or the majority, and we're going to be visiting that to see in the future what's going on. So it's really exciting. Um,
That is so exciting. I can't wait to hear what you all find out. And it sort of reminds me of May Wan Ho's work and writing about the nanotubules in fascia and in our connective tissue. And I wanted to kind of ask you about your work with easy water, exclusion zone water, and how that applies to the fascia, to joint lubrication and swelling, to muscle fibers and the way things are working on that level. I know you, like you said, you got your start working in muscle fibers and contraction. And I know that you speak a lot about that easy water and how that might facilitate things when we're talking about joint lubrication and swelling and some of the things like that. Can we talk about that a little bit?
Yeah, it's all essentially all water, pretty much all water that's exists in our bodies, and it's not just the intracellular water that we're talking about, but also the extracellular water, and you're probably aware of well, I think his name is Jean Pierre Gumbarto, or yeah, who showed realistic situation that there's actually flow in fascia. You could actually see little droplets that are propelled along, and so it's pretty clear that there's water that's outside. It's almost certainly easy water, and it actually can flow along. So it's clear, and also it's clear that various phenomena helps to flow that. And so, for example, one so one phenomenon is putting your hands on it, on the fascia, for example, and your hands even gently put on the surface. uh, uh, there's a lot of infrared energy that's being transmitted to the region. And so that energy, the infrared energy, easily converts into flow because just in the same way that the water flows actually into a tube, the same thing happens. So that's the infrared energy is being used to build easy water, which in turn propels flow. And by the way, the same thing happens for example, in xylem and tubes that run up all the way up to the top of 300 a 300-foot redwood tree. It's really quite amazing, and so we think we understand it's the same phenomena as what happens, for example, in the springtime, because the springtime is filled with easy water, right? Because there's a lot more infrared energy, and this infrared energy now begins the flow into easy water, which separates the positive and negative phenomena, which then propels the water that's up into the tree. So there's a whole lot of phenomena. It's not just the fascia, but throughout this kind of phenomena that's all around. And
Absolutely. And we have sort of a different, what we learned in school is that you have these cells and they're floating around in this water and the cell. You go in the cell, there's all this free space and you can float over to the mitochondria or the ER or, whatever it is. But actually, that's a misnomer, right? We are, if we could do that sort of magic school bus cartoon and shrink down and go into our body and check it all out, we'd find that it's extremely crowded, right? We only can get a few water molecules away before running into another structure, right?
totally, we if you if you consider the results that have been accumulating over the past 100 years or so, it turns out that the spacing between um, between solid surfaces inside, it turns out it's equal to roughly 7 or so molecules lined up. That's all, 7, 6 or 7 or 8. And it's really a tiny amount that actually is so if you look at electron micrographs, actually quite a few electron micrographs, it looks like a jumble. It's I mean, so phenomenal that there's almost no space with 7 or so lined up approximately on average. It's extremely crowded in there. And to think about what happens during um, it's almost impossible to think that various molecules can actually move back and forth in this. The matrix is incredibly difficult that everything is around. And so, many people think of diffusion and osmosis and such and moving around, but it's just impossible like that for happening. And so you begin to think what actually does happen in certain cases, and it ain't the way we learned with diffusion from one place to another, like enzymes and such. It simply doesn't happen. So there's more to it. We can talk about it if you want, but I think you've got more to talk about, I think.
Well, I'd love to hear more. And this is really sort of, quantum biology kind of came up in the 50s. Schrodinger talked about it at the turn of the century, but the things that came out in the last 20 years out of Berkeley and MIT are really showing that these very small oscillations, these very small actions can really power what we learned in school, right? There's something else deeper happening. So if you'd like to add to that undercurrent of
Uh,
biology that's being powered by that, please.
but yeah, so um, I guess I would like to touch um, on the area of consciousness, and you like that issue, and we're beginning to study that right now. There's there's a lot that's taking place now. We actually um, we actually I organize each year. We we have we have a group of people. We get together with something like 200 people or so every year in right near Frankfurt, and there are a lot of presentations each year. It'll be in August, in October, or approximately the middle of October, and there are lots of presentations each year of people who show demonstrations um, about the idea of memory in water, information in water. And it all starts, I guess, with the most famous, the Emoto people, who I know I'm connected very well. And for those people who know about it, but I think a lot of people don't know, and they put attention, especially people like Emoto, puts their attention into water and then freezes the water. And if the intention is put into a positive concept or vein, and then he freezes the water, and he finds out that there are beautiful crystals. And at the same time, instead of doing that, he thinks of ugly kinds of things, negative emotions and such, and freezes. And according to that, what the crystals that he forms are actually ugly crystals. So a book was produced more than 20 years ago, and it hit the market. It was tremendously phenomenal. However, in terms of the science, this is not a way that science is done, because what he would do is he would freeze he would freeze 50 petri dishes, and he'd put his attention to maybe 50 of them, and he'd cherry pick the one that chose the best, and the one it turns out to be the one that demonstrates best, the one that showed what he wanted to show best. And so, a lot of people don't pay a whole lot of attention when they put their minds to it, because this is not the way the science works. Now, more people are beginning to do these kinds of experiments, and they're demonstrating that that by repeating the experiment, actually there's some information coming through that demonstrates that this is for real. And so is beginning to come. There are several people who are doing experiments. We're waiting for them to come out. Now that's at one end. He's a spiritualist. He was a spiritualist, and he passed, I think, 6 or 7 years ago. But then at the opposite end are the works of Luc Montagnier. And but other people may be less familiar. And he happens to have won a Nobel Prize. Unfortunately, he passed about 3 or 4 months back, 5 or 6 months. In fact, just this weekend, there's a memorial service in his honor. He won a Nobel Prize for discovering or identifying HIV. And he started doing experiments. And, these are when the kinds of experiments are done by someone who won a Nobel laureate, you pay a bit more attention to it. And his result is phenomenal. One of the main results is this. So he would take some strand of DNA, say 100 base pairs, and he would dilute that into water, and he would put this into a sealed container, and impossible for water of any sort to leak out, very thoroughly sealed, so no chemical contamination, so to speak. And nearby, say a foot away, is another container that's full of water. And he contended that what happens is there's information that's coming from the tube that's coming out, the sealed tube is somehow transmitting information to a nearby tube, and throw away the original, and you've got this substance, the water that contains information. And in order to demonstrate that that's true, he would take that, and using the PCR method, the same one that we use all the time right now, he demonstrated through the PCR technique that after amplifying the information, he showed that the new DNA that came had the same sequence as the original, original sequence of DNA. And it rose, a lot of skepticism arose from many people, but there are now 3 different laboratories that apparently demonstrated the same result. And so here you have now the opposite end of the spectrum. You have people who have been able to confirm what he had discerned from these original experiments. And Luke would come to our for the past decade or so every year and presented more of the same, until several people repeated, and unfortunately he passed on. So what I'm trying to say is that from one end, from a spiritualist, this is Emoto, all the way through many people who had different kinds of evidence, all the way to Luc Montagnier at the, you might say, top of the pyramid, top of the pinnacle of having won a Nobel Prize. There's evidence. So to summarize the whole thing, there's now a lot of evidence that somehow consciousness or subtle energy can be imparted to water. And I guess I should end there because I don't want to go on and on, but this is terribly important. And there's now enough evidence to suggest that this is really a frontier subject, that people understand that this looks like it's real. There are many people who have evidence that this is real. And this is going to be, I think, a frontier study among others to discern more about what's going on in the water, including studies from our own laboratory. I think I should stop there.
Absolutely fascinating. I can't wait to hear the results of those. That's Luke's work was just is phenomenal and really takes us into opens up a whole new window into what our biology is doing, what powers it, how do we transfer information. And that's really what, comes up for me when I'm studying your work and their work. And water is this vast communication network that we have that spans throughout the globe, throughout the universe. It's really phenomenal and mind-blowing, and something I can't wait to hear about more with the research that's coming out of your lab. So I know that you are an extremely busy professor, researcher, educator, author, all of those things. I really appreciate you taking your time to talk with us today. Before we end, um, can we talk a little bit maybe about your top 3 ways to support this fourth phase of water, both intercellularly, extracellularly, all of that? How do you have some favorite ways to practically, practical tips to help some of our listeners take steps in supporting that fourth phase of water?
Um, well, your statement is sufficiently broad that I'm not sure exactly what to say, but let me just say one thing. We've managed to well, let me back up a step. Our cells are filled with easy water, and what we found in the 2 books that you're mentioning, the Cells, Gels, and the Engines of Life, and also now the fourth phase of water, which has become really popular and translated into, I think, 10 different languages, that talks about the role of fourth phase water in health. And the one phenomenon that we've come to appreciate is making sure that our bodies or our cells are filled with easy water, because the easy water is central to, according to the evidence that we have, is central to so many phenomena that make us the phenomena to work. You need to have a complete amount of easy water. Otherwise the cells are not able to perform the way that we think is helpful. So it means that what we think about in keeping healthy translates in a very simple way to creating enough easy water. If we don't have enough easy water, then our cells are not functioning. So there are a half dozen ways that I could enumerate that maybe we don't have enough time to very simple ways for us to create easy water. And they're not complicated, they're simple. And so that I guess, if we had enough time, I could talk about them there, but they're essentially, I'll just mention one of them, and because there are half dozen or more, or many drink more water, because the water that you drink, some of that gets turned into easy water, which is what you need. So you need to be hydrated, and that you can create that by drinking enough water. And another way, just out of a half dozen, I'll just mention, in saunas, or saunas, as the Finns would call it, and you go into this really hot room, whether it's dry or whether it's moist. It's just full of infrared energy, and infrared energy creates EZ. So many of us, you just go into a room that's filled with this kind of heat, water, infrared energy, and you come out 30 minutes or 20 minutes later feeling good. And I think you feel good because the infrared energy is building easy water, and therefore in building the easy water, you just immerse yourself in that heat, and you feel better when you come out. So there are other simple ways that you can create that. So let me then, there are half a dozen simple ways to do it. So this is number one, the creation of easy water. Another phenomenon, I'm thinking out loud. It has to do with health. And if you read the textbook, I think you're going to be misled. There are many things that appear in textbooks that I believe are simply wrong. And they're simply wrong because they rest on a foundation, the standard foundation that you can read about. But if you accept what we and others come to some sort of understanding, especially about the water, about the role of easy water, and the different, shall we say, different forms of energy that we get from our environment, and different phenomena that extend from that. There's so much of it. In essence, so many mechanisms. If you come to accept and believe the evidence that we argue is the truth, then you arrive at different mechanisms that are different from what the textbook teaches us. So this would be a second point. And it's not just a few trivial ideas. It's more than that. It's just all over the place. So, I hesitate to say it because it sounds arrogant. But I think that so many phenomena that stem from this kind of new understanding based on water, it leads to understandings that are completely different from what you find in the textbooks. Okay, so that's the second point. Third point, there are so many things I'm not sure about. So the water, the concept of water extends beyond just biology. I mentioned a few moments ago, for example, about the water that goes all the way up to a 300-foot tree, to redwoods and such. And there are phenomena that go way beyond that, in which this kind of new understanding or fresh understanding about water and the role of water extends to phenomena throughout nature. It's not just in biology. It extends beyond biology. And I have a book that is essentially almost completely done. I'm waiting for my son, who's the artist who illustrated the fourth phase of water. And this guy, my dear son, who is a wonderful artist, he's the one who illustrated the fourth phase of water book. So many have commented on it. He's really gifted, but he's busy reconstructing his home, he's remodeling, and I'm waiting for him to finish with a few, just a few illustrations that he needs to work on, but he's very busy and his family would like him to finish. So it's waiting. And there are so many phenomena that extend not only from the idea of easy water, but details about how things really work. And I'll just touch on one of them because there are many that are discussed and described in the books. One is weather.
Mm.
4 chapters appear on that. And one of the things is that nobody understands weather. If you think about, think about from the beginning, from the start, and think about asking a question. It's just you're dealing with water, and the question is, well, how do you explain starting from a viewpoint of water, which is what weather is all about? How do we explain it? And that involves evaporation. It involves cloud formation. What turns on the rain? We have lots of gray clouds. Who is the guy who unzips the clouds to let the water come out? And by the way, what keeps the clouds up in the air? If you were to climb a ladder with a pail of water and dump that water from way up there, it will come down and it can wash your hair. But why does that occur? And why does it sometimes happen and sometimes the gray clouds remain but don't unleash that water? And why is it that the water comes down in droplets instead of like a bathtub full of water, which is what you'd expect for a change of phase from a water that consists of a vapor somehow, which comes down in one droplet at a time instead of a whole dump? Why is that? Well, nobody understands that, but yet it's really important to understand it, and people don't understand. So I guess the third point that I wanted to mention is that there's so much that is centrally involved in phenomena that relates to the fourth phase of water that people need to know. And so many of these phenomena underlie phenomena in nature that go way beyond just health. So that would be the third point that I would end up with. I'm sorry, I've just been blah, 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 blah. So those are the 3 points.
Well, we greatly appreciate you and your blah, 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 and, really explaining all of this to us. It's absolutely phenomenal. And I agree that once you start looking at the evidence, it's really compelling, the picture that's being painted. And, for those of us interested in quantum biology for health and for wellness, it really opens up a whole new paradigm, a whole new perspective on what's powering our life in living systems, and really the relationship we have with the world around us, and how it's inseparable to that powering of living systems, how we really don't end at the barriers of our skin, and we allow for this beautiful interchange of energy with the environment around us. So I really, really appreciate talking with you today. I know our listeners have really appreciated hearing what you have to say. I do want to tell people listening today on the summit that you can check out Jerry's speaker page. It has links to his books, his research, ways to donate so that this phenomenal research can continue. Do you have any last parting words for us? Anything that we should be checking out or following you on?
Yes. How can I say no? There's actually so much. Thank you for the opening. I guess I just want to say in capsule form is that all of us need to keep our minds fully open to the most fundamental phenomena that exist around us. I think that it's been difficult um, the difficult to uncover fundamentals. We start building foundations. We start building on foundations that we come to accept, and yet few of us try to dig beneath these acceptable, so-called acceptable foundations. We we presume that at certain levels of foundation, we come to accept those foundations. And often we accept those foundations because they come from towering scientists who revere as people who have created those foundations. And, we don't exactly presume that those towering scientists are necessarily right. They eat the same food we do, they make love as we do, they go to the toilet the way they're human beings. It's they might not be right. And I just want to mention one typical, one representative example, and that is, we accept pretty much the idea that the foundations of the structure of the atom, for example, foundationally must be correct. It's been 4 or 5 generations. The great Niels Bohr started with a sort of solar system model, and that has been supported by the great quantum mechanical scientists, Edward. But how do we know? How can we be sure that those concepts are actually correct? We assume that they must be correct, and we assume that all the, for example, quantum mechanical contributors are right on, and there are many contributions have been necessary to make those concepts really work. And by now there are 60 or so fundamental subatomic particles that are necessary to make it all work, to keep it put together. And is that something that we should rely on, or are there questions about this? And I'll just make two specific points about that. So the first one has to do with the nucleus, right? Okay, so we know that the nucleus is still considered packed with protons and neutrons, and the neutrons are neutral, but the protons, are packed together so tightly that you got a whole lot of positive charges put together. Now they repel each other, or at least our understanding is that they repel each other. So you'd expect the nucleus to explode, because what keeps those particles from exploding? This is you might say, is a kind of middle school concept, all positive charges stuck together. Why doesn't the nucleus blow up? And the physicists have managed to respond to that one, and they said, well, there's the so-called strong force. Okay, and the strong force is kind of like a Band-Aid to cover up a gaping wound. It's the so-called strong force that has exactly the right characteristics to counter this explosive force. That raises some concerns. Is there any evidence that this special strong force really exists? And then there's just one more. We're told, again, middle school stuff, that if you have a nucleus that's positively charged, and surrounding that are electrons, and specifically orbits or orbitals, right? But they're negatively charged. So these orbitals are responding to negative charges, and the nucleus consists of positive charges, and we're led to understand that pluses and positive charges and negative charges are actually attracting one another. So how come the atom doesn't collapse? So there's just two examples about fundamentals of science. We have to actually ask the question: are these fundamental considerations, are these something that we can take as fundamental truths or not? We have to look back and see. So I'm sorry,
Absolutely.
I mean, blah, 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 but we need to really consider it, whether those concepts are really, really okay. I'll end with, yeah.
Absolutely. Jerry, you are speaking to my heart. The second time after flunking organic chemistry, my professor said, just give me the answer that I want.
I,
Don't
I got you.
enough with your thoughts. Just
Yeah.
give me the answer. And I am loving quantum biology and learning about some of these different aspects. But then I always get to a point where, people are saying, "Oh, you're so into quantum biology," and it's like, well, until we figure out the next step in the ladder, right? It's a continual thing, and the thing that drives us is that curiosity, is that ability to step outside of the box and say, maybe atoms don't work like that. Maybe electrons don't orbit. Maybe it's more of a cloud, or, all of these different things. That's where science comes from. That's where advancements in knowledge and treatments and all of it. It all comes from that curiosity and that willingness to keep an open mind. So I deeply appreciate you as a scientist, a professor, a researcher that is continually doing that. It has had a profound impact. Just that attitude, just that perspective has had a profound impact on my life and the way I approach science. So I really, really appreciate your approach, your open-mindedness, your willingness to entertain things that are outside of the box because they might be true. So I, I really, really appreciate that. And I really appreciate you taking the time to speak with us all today.